I was going to introduce myself, but I think all of you have done a great job. I just want to add a couple of things. I was born in Mumbai. I was raised in Mumbai, and I went to what was then called King George High School, which is where I met Avi. So Avi, amongst all the people here, I've known him the longest. Then I went to Ria College and then to IIT Hostel 3. And so a lot of the IIT people here, I know from that time. Some of them I haven't seen since I graduated. So it's really nice to at least remotely meet all of you. Let me tell you a little bit about the kind of work I do and people in my lab do. Mostly, we're interested in taking images. And for us, it's mostly some form of biological images. And then we try to understand what's happening in those images. We try to quantify them by doing some kind of mathematical computational analysis. So here's an example of a project we work on. This is a project where we try to understand how Parkinson's disease develops in the brain. So we look at images like this. Can you guys see my cursor? Okay,
yes, we can.
Yes, we can. Yes, we can.
So here's an image, for example, of somebody's head, and the head is in this position. The nose is down here. The eyes are here. This is the salivary gland. And then we try to understand how the disease actually affects these people and how the disease progresses with time. It's quite complicated because Parkinson's disease is not a homogeneous disease. Different people follow different trajectories of the disease. So it's really useful to understand what these trajectories are and how the disease actually differs from person to person. We also work, so we're operating, these images, the Parkinson's images, are at the scale of a millimeter. So 1 pixel, the smallest piece of information in there is 1 millimeter thick. That's in contrast to the image on the right there, which is an image of a single molecule. So this is a single protein, and there the resolution is down to a few angstroms. And just to give you a sense of what an angstrom is, so the hydrogen atom, which is the smallest atom we know of, has a diameter of 1 angstrom. So these images, which are on the right there, they have a resolution of about a few angstroms. So we're looking at really very high resolution structures of proteins. And the COVID-19 research that I'll talk about in a second relates to that. So it relates to understanding the molecular structure of certain proteins in that virus. And these proteins are really important in terms of how the virus actually works. We do a range of research. Some of it relates to basic science. So the molecular structure part is basic science. It involves trying to understand how things work rather than actually developing a drug. The disease part is a little bit more applied. So I thought I would tell you guys something about how we did this while we were in a pandemic. But to explain how we had to do this, I have to tell you a little bit more about what the actual research was on SARS-CoV-2, which is the virus that causes COVID-19. And I think maybe you guys would find this interesting. And then I'll talk about how we did it, what some of the challenges were, and what I learned personally as somebody who was trying to run this lab from this whole effort. Okay, so I want to tell you a little bit about the virus. So the virus is called the SARS-CoV-2 virus. And I should explain, I'm not a biologist or a biochemist. So this is just stuff I've learned from talking to biologists and biochemists. The virus, for its genetic material, has RNA, which is inside this membrane. So this whole thing is the virus. To give you a little bit of a sense of how big this is, the virus has a diameter of about 100 nanometers. And, I see this thing is hidden back here. To give you a sense of that scale, how much 100 nanometers is, the diameter of typical human hair is about 100,000 nanometers. So we're talking about something that's 1,000th the size of the diameter of human hair. You can't see this virus in an optical microscope. You need an electron microscope. That's how small this is. So, so the virus has RNA as genetic material, and that RNA actually sits within a membrane. There are several protein molecules that go across the membrane. These are these membrane proteins. The one that's interesting to us is this protein called the spike protein. And it's actually a trimer. So what that means is there are 3 strands of proteins that come together like this, like 3 fingers. And this has a length of about 10 nanometers. So if you actually take an image of this virus, so this is an electron microscopy image from NIH. This is the National Institute of Allergies and Infectious Diseases. This is where, this is the institute that Dr. Fauci heads. That's what the images of protein look like. And this fuzzy stuff you see around the protein is actually the spike protein, this thing. And it's because of this fuzzy nature that this virus is called the coronavirus. So this looks like a corona. The word corona means a crown. So it looks like a crown around that sphere. It also kind of reminds me of the solar corona, the solar flares that come off the sun. But the fuzziness is due to these spike proteins. So what happens when this protein infects your body? So the mechanism by which this protein enters your cell depends on the spike protein. So here's an image, again, an electron microscopy image of the coronavirus interacting with cells. And you can see most of these interactions have to do with the spike protein. And the way this works is that in the membrane of your cell, there is a protein called the ACE2 protein. And the spike protein then attaches to the ACE2 protein. So there's a piece of the spike protein that's movable, that's flexible. And because of its flexibility, it's able to attach to the ACE2 protein. And whenever this attachment happens, that triggers a whole set of reactions, which causes the virus RNA to enter into your cell, to duplicate itself, and then spread away. So this piece is really critical in the infection part of the protein. Okay, so that's the piece we want to understand. So our task was sort of a basic science problem of trying to understand how this protein, how the spike protein actually changes, what its flexibility is like, and to try and actually measure it from individual spike molecules in a solution. So here's the whole sort of the whole scientific pathway by which we try to understand this. So the way this works is, first somebody takes a whole bunch of coronavirus particles, purifies the protein in solution. So this is not the spike protein, this is some other protein. This is just an illustration that Gabriel Lander made during his PhD. He has a lovely YouTube video about this whole process. So if you're curious, you should try and Google it. So you purify the protein, and then through a number of steps, you try to get that protein in a thin layer of water, which you then freeze at really low temperatures. So what you end up with is a thin layer of ice in which these protein molecules are stuck at different orientations. So they are stuck at random orientations in that ice layer. You then take that and you insert that into an electron microscope. And when you turn the beam on, what you get essentially are shadows of that protein. So this whole process is called cryogenic electron microscopy done at really low temperatures. And what you get are images like this. So these are, as I said, the shadows. I'm using the term shadow in a very loose sense, but they appear like they're shadows of the protein molecule. And then what our task is to take these shadows and then reconstruct from it the actual 3D structure of the protein at as high a resolution as we can. And this image is a little bit deceptive in the sense that that's a really good image. Usually the images we get from the electron microscope are extremely noisy. You can barely see what the protein is. And the contrast in these images depends a lot on the size of the protein. So the bigger the protein, the larger the atomic weight, larger the molecular weight, the better the contrast. So these spike proteins are really tiny. And we get images that look like this, which are really, really noisy. So that's the computational challenge. The computational challenge is to take these images and from that reconstruct something that's reasonably high resolution and meaningful. The one advantage we have, the real ace that we hold in our hand, is that we have a lot of these images. We have 66,000 images of these proteins. And so because you have a lot of images, there's some hope of combating noise. I should say one more thing, which is this whole process from purifying the protein all the way down to creating a structure takes a lot of work. We don't do all of this. We come at the end of the step. So somebody gives us images, we do the computation, and we try to create structures. But this process of the biochemistry and the electron microscopy is actually extremely challenging. So we're kind of just the last chain in this long chain of research. And a lot of people contribute to this. And in some sense, I think, mostly, I guess, because I do computation, I think the biochemistry part and the microscopy part is at least as challenging, if not more challenging, than the computational part. Okay, so that's the problem we have. We want to take these images and try to then understand how that spike protein moves. We want to understand its flexibility. Okay, so before I show you what we actually got, let me tell you a little bit about how we did this. So in Connecticut, which is the state I live in, our lockdown started on March 20th. So March 20th, the governor of Connecticut said, everybody now has to stay home unless you are an essential worker, unless you are actually a physician or a nurse or somebody who works in healthcare. So we all went home on March 20th. And things have actually been slowly easing up. So May 20th was what we call the phase one of the lockdown, where some additional people were allowed to go back to work. And recently, October 8th, we had the phase 3 of the state reopening. So phase 3 simply means restaurants can now open at half capacity and so on. We are still working at home. So I haven't seen any of my lab members or students or postdocs since March 20th. So before March 20th, we were working on other proteins. We were also working on other problems like Parkinson's. On April 8th, we pretty much dropped everything else in the electron microscopy field and started working on this spike protein. By July 23rd, we had some results. We submitted our paper, and just last month, our paper got accepted. So this entire span of research was completely done offline without anybody physically meeting anyone. So there were a lot of challenges because of this timing of this research compared to the pandemic. There were also space challenges. So we of course cannot do all of this. We are only the computational part, the last part. So this was a collaborative effort that was in some sense an international effort. So I'm located here in Connecticut. We collaborate with Jose Maria Carazo, who is in Spain. He runs one of the big European electron microscopy centers. And this also involved working with Jason McClellan, who is down here at University of Texas. So to tell you what everybody did. So Jason is the person and people in his lab purified the protein, did all the electron microscopy, created the images. Those images then went across the Atlantic to Jose Maria's lab, who reconstructed stationary high-resolution structures for these spike proteins. And then Jose Maria, who's a good friend of mine, got me interested in understanding, in trying to calculate the flexibility of these proteins. So what's remarkable about this is that I have never met Jason McLennan. So this is all Jose Maria trying to coordinate a research effort amongst labs that haven't worked before. So that was also a challenge. Okay, so back to the spike protein. So these are images that we got from Jason McLennan. And here's what Jose Maria was able to do. So he was able to actually reconstruct from those images fairly lovely high resolution structure of the spike protein. So this structure you see, imagine that it's one of these proteins standing upright. And he was able to get a resolution of about 3 angstroms, which is quite remarkable, which means all of the details in that protein that have the size of 3 hydrogen atoms or larger, he's able to see in spite of all the noise in the images, which is just astonishing, I think, that he was able to do that. So then he sent us a whole bunch of images, and we started looking at how that structure actually changes. So there are 60,000 images. Each of those images has a single spike protein in it. And so some of the spike proteins have structure in one conformation. Some of the spike images have structures in other conformations. So we have some methods for trying to understand which of the images have structures in one particular conformation or another conformation. And we found that there were actually several modes of change of that structure. So here's one mode of the change. You can see what's happening is if you concentrate on this piece, so think of this as your thumb sticking out and wiggling, you can see that the angle of this with respect to that is changing. And that's the most predominant change in that spike protein. And very likely that's the change that might be involved in the binding of that protein to that ACE2 enzyme in the cell membrane. We also found that there are some other modes which appear to be quite mysterious. So here's one we found. So here's a mode in which that density of the thumb seems to disappear. And other people who have studied this have also found that something like this seems to exist. And we don't clearly understand what's going on. Is this a more subtle motion where the thumb is becoming maybe less dense? So we cannot find its structure above the noise level, or maybe something else is going on. I should say something about the more global effort to understand this. So what happens with these structures is we reconstruct them out of these very noisy images. And so a big issue in all of this research is, how do you know that what you got is actually correct and it's not the effect of noise? And so each lab that attempts to do this, we have our own internal sort of measures to check that we're not doing anything wrong. But more globally, different labs which have different techniques, when they start producing similar results, we have some guarantee that, okay, we're not doing something crazy. So other people now who are studying the spike protein are also finding something like this, but we don't clearly understand what's going on. So there seems to be some very interesting movements of that protein. The next step, which now people are beginning to do, is to try and understand what happens when antibodies are attached to this. So an antibody is something that your body releases, that then there are antibodies that target the spike protein. And our guess is that possibly one mechanism by which the antibodies work is that they might prevent this motion, or they might prevent the kind of motion that allows these proteins to attach themselves to the ACE2 inhibitor. But that's the next step, and a lot of people are working on that now. Some of the vaccines that are coming online, the Pfizer vaccine, for example, targets the spike protein. So this stuff is pretty important. This stuff is a basic science that tells us how the spike works. And the spike protein, incidentally, is not just present in the SARS-CoV virus. There are other viruses that also have the spike protein. Uh, so this is pretty central to understanding how the virus works. Okay, I want to mention members of my lab who were involved with this. So Brent Foster, who's one of my postdocs, Jose Luis Villas, who's also a postdoc, worked really hard. Brent especially worked incredibly hard trying to do this. He worked over weekends, he worked at home, we Skyped during all kinds of hours. And a lot of the success is due to his time and effort on the project. Okay, so what did I learn running this during COVID-19? So I think the most important thing I learned was something that I guess I sort of knew, but it came back home again very strongly, which is that the motivation to do something, kind of the non-scientific motivation, if you will, really plays a key role in getting people to go beyond their comfort zones. So most students and most of my postdocs seem really motivated by an application they can relate to. Some of us, there's a small percentage of students who will follow a subject because they're interested in just the subject itself. Most seem to be really motivated if you can relate that piece of mathematics or computation to something in real life that they can relate to. And a social purpose can be really motivational. So I see that because I'm really fortunate that I work at the interface of biology and medicine and computation. And so it's not too hard for me to find some kind of an underlying social purpose for the research we do. So many years ago, here's another example. We worked on a problem which was trying to detect preeclampsia. So preeclampsia is this disorder that pregnant women have, which if it is undiagnosed and untreated can cause death. And it's among the top 3 causes of death during pregnancy. For a few years, we worked on trying to develop an app to detect preeclampsia. And you could see the same thing. The students were really motivated. They put in far more time than I expected that they would have put into this project. So for all of you who teach quantitative analysis, one of my suggestions is maybe because you're a professor, certainly this is true for me, that I can sort of follow a subject up to a point because I'm interested in the subject. I'm just interested in the logic and the ideas of that subject. But for a lot of students, actually relating that to some application in real life is hugely motivational. There are some problems, and I'm not entirely sure how to solve them. But for those of you who are attempting to either teach online or run a lab online, maybe this will give you something to think about. So video collaboration. So this whole research was done by me sitting in front of Zoom and talking to my postdocs and students. But there are some problems. The biggest problem is that not everybody has access to bandwidth. So there are, even in the US, we have postdocs who live in apartments and get access to the web is by Wi-Fi. And so, there are times in the evening where all the apartments are on Wi-Fi at the same time, and you just don't get the bandwidth. And so the variation in resources is the big issue. And in particular, what that means is that our meeting schedules become very uncertain. So it's very hard to say, we're always going to Skype or we're always going to Zoom at 10 in the morning or 11 in the morning for an hour, where we have sufficient bandwidth to look at the results and to discuss a few things. So our schedules become really uncertain, and that's a big issue. The other big issue that came up a lot was um, we lost a lot of the informality. So, and this is really important during research. So we find that, I find especially that when I have a lab meeting in the university, when everybody's sitting around a table, it's very easy for me to look at people to see whether they are confused, whether they have a really interesting idea they want to talk about, whether they are enthusiastic or not enthusiastic. That's really difficult to do over Zoom. And I don't really know how to overcome this uh, other than to just do more Zoom meetings so people get used to it, which is not a great answer. The second big issue we have is because we do a lot of quantitative analysis. When we are in the university, when we're doing this in my office or in my postdoc's office, we all have blackboards and whiteboards, and we walk up and we write informally equations or we draw diagrams and things like that. And those are really difficult to do online. So I wish there was a better tool for that than using the Zoom whiteboard, which I find really difficult to use. And the last thing is that brainstorming is really hard. So when we're in the office, we just sit around a table and you have your cup of tea or coffee or something like that. And when you're thinking about something, very often there are these long pauses. You just, you stare at something for 5 minutes before you have something interesting to say. Just dead online video for 5 minutes is really difficult to stomach. For some reason, it's easier to do this in person than it is to do while you're on Zoom. And we still don't have a good answer for this. Okay, so my suggestions from this experience is that it's really worth your while to actively work towards keeping all the informal relations with your collaborators, with people in your lab, with students and postdocs. When you're in an office, it's very easy to just walk up to somebody and ask them, what do you think about this? Or what are you working on? It's much harder to do that when you're online, when you're not in the office. But it's well worth the effort to actually keep those informal relations going. Because I think a lot of the ideas, the new ideas or solutions to problems come from these informal relations. Try to meet regularly with your lab members, and if you can do that virtually, maybe that's the only choice you have. So we try to meet at least once a week for about an hour. Initially, when we started this back in April and May, a lot of those meetings were very formal. People treated them as if they were in class, but they have become somewhat informal over time. And I think the thing I miss the most, and it probably maybe one of the most important things, is to keep the community of your students and postdocs and labs and even the larger community of your department engaged in some kind of a communal activity. So we still feel like we are part of one organization, that it doesn't feel like we're just some consultant sitting at home, giving some ideas once a day or something like that. I try to really work hard towards keeping this community going. And at least for me, that is very meaningful. So I want to see my students, I want to see my postdocs, I want to know what's going on in their lives other than just, they're doing research. And I want to know that they're okay. Okay, that is pretty much all I have.